So title this simply Singing in the Brain, but you can also say speaking in the brain. And particularly, we're going to talk about the forebrain, the parts of the brain that are involved in complex learning and memory. And talk about species that have the ability to produce imitated sounds, like some of the ones shown up here, and some close relatives that don't have that ability. And for those that know, this is Jennifer Lopez up here singing alongside with this parrot here. And I have broken my talk down into three parts in terms of the biological level of analysis: the behavioral, the anatomical, and the genetic. And then I'll end off with a new hypothesis we're trying to test actually in mice with their vocalizations. So starting with the behavioral, and what do I mean by vocal learning, as you heard before. And if I don't cover something here, if you want to see more or learn more about this, I encourage you to go read an article that I try to write for a general audience called Evolution of Vocal Learning and Spoken Language in Science magazine that was published last year in a special issue. So when we humans think about spoken language, we tend to think of it as a unitary trait that has, it's all as one big package. But actually, it consists of a complex series of multiple traits. And some of those traits highlighted in blue here are traits that are found in lots of different species out there. Not all of them, but lots of them. And some of them highlighted in red here, like vocal production learning, is rarely found in humans and a few other species. And with this idea of spoken language, or with this breakdown of the components of spoken language, I see it as a specialized form of learned vocal communication with some components found in most vertebrates and a highly specialized vocal learning component found in only a few species. And so let's look at those components and what are those species. There are 5 groups of mammals that have this vocal production learning. Us humans, of course, and the cetaceans. Those are dolphins and whales, bats, elephants, and seals amongst mammals. And there are 3 groups of birds: parrots, songbirds, and hummingbirds. And many people already know that parrots have the ability to even imitate some of our own speech sounds. And so all of these species have this ability to produce novel sounds that they weren't genetically encoded, but they all have close relatives that do not have this ability, like chimpanzees are to humans or falcons are to parrots. So it's been argued that these 5 groups of mammals and these 3 groups of birds have evolved this trait independently of a common ancestor. What about another component, auditory learning, or what I call also comprehension learning? Well, this trait exists in many of our pet animals. Like you can teach a dog to understand the word sit in English, or siente se in Spanish, or Swahili in Japanese. Continue humans, roll over, get the ball, and so forth. But in the auditory comprehension domain, a dog can't say, okay, you got it, I will sit or roll over. Instead, a dog goes woof. And it can learn how to produce woofs in different contexts, like for food, for attention, and so forth. But it actually can't say, I want food, which would be vocal production learning. So now, not all vocal learners are equal in their ability. Of course, us humans are the most advanced, but even amongst the non-human vocal learning species, they vary by degree. And parrots are amongst the more advanced non-human vocal learners. And here is an example.
Never shake a baby bird. That would surely be absurd.
Never shake a baby bird. That would surely be absurd. Are you not a crook? My name is Tesco. Again, meow, meow. Give me a kiss. Hey, kid, has cheeseburger. Are you a little stork or a stormtrooper? Disco, I used to be a spy. And a parakeet. Sure, I'm a doctor, not a parakeet. I did, I'm a doctor.
So that's pretty amazing. And what Disco is doing here is not only having auditory learning, but it picked up words from another species, humans, and actually imitated them in a manner that Disco is actually having a conversation. It's not just putting out 1 or 2 words at a time, but it's whole sentences. And roughly 70% of the time, Disco will recombine words that he's learned into meanings that actually his listeners don't know what they mean, or why there's a random combination. But about 30% of the time, actually combining those words in appropriate context even individual words, like when it sees the dog here and goes meow, kind of gets the actual individual species wrong, but the actual category correct. And so, so that's quite amazing. And then every once in a while, a study comes along where one of our closest relatives, in this case, Koko, a gorilla, has been argued to actually learning something that's starting to approach a speech. And people get excited about it, but it's not what you see in these advanced vocal learners. And here's an example.
How about when you're coughing? That was good. You did a sneeze and then a cough. Excellent.
Yes, that was excellent, but it's not the same as what we see in these vocal learning species, where it's going on here is that Koko has good auditory learning skills and understands these human speech words, but Koko has limited ability to modulate the sounds or the air going through her larynx. In this case, with a cough sound, can blow up air voluntarily, but actually can't modulate that airflow through the larynx in the way we humans can to produce novel sounds. And Koko actually does have some kind of sign language skills, could sign over 1,000 words and learn several thousand words auditorily. So it's the limitation of the production more than the perception. Before I go on, I just want to make sure that you guys are hearing these videos well. Is that the case?
Yeah, we can hear everything.
Fantastic. All right. So so this has caused myself and other scientists to say, ask the question, how come we can't get some of our closest relatives to say something simple as the word apple, where those who are more distantly related from us, not just 6 or 10 million years from a common ancestor with us, but 300 million years from a common ancestor with us, like these parrots, and can imitate many different varieties of apples, like Golden Delicious and so forth. And this is the question that I've been asking for a number of years now. And I argue it's in the anatomy of the brain. And that anatomy is explained by what I'm calling now the motor theory of vocal learning origin for the second part of my talk here. But my theory here is not the only one that's out there. There are multiple hypotheses that have been proposed to explain the differences between those who have this ability to imitate sounds and those who don't. One is that there's a general difference in the forebrain. The green here is indicating the cortex of the brain. The red is the brainstem motor neurons that control vocal behavior. And the blue is the actual muscles themselves that produce the sounds. And so one argument is that the areas of the cortex are bigger, like the auditory cortex or Broca's region, and the whole brain is bigger, let's say in humans, relative to the body size, and this explains the difference. The other is that it's not in the brain itself, it's actually in the vocal organs, that humans have a more descended larynx, and this allows us to produce a greater variety of sounds that other species can't produce because their larynx is too close to the opening of the airway. Another is that it's not a difference in the peripheral organ, but it's a difference in the presence or absence of a neural circuits, the haves and the have-nots. And if you have the circuit in the forebrain that controls the vocal muscles, then you'll have vocal learning. Another hypothesis is that it's not a have or have-nots, it's one of degree, that it's there, but it's either has what we call a direct projection to the motor neurons as opposed to indirect one with these arrows here that control the voice, or it has a direct projection in all species, it's just more enhanced in the vocal learning species. Another hypothesis, it's not anything to do with the connections of the brain to the peripheral organs, but it has to do with something inside the brain itself that is a more direct projection from the auditory regions to Broca's area for speech production, or an enhanced direct projection. Or coming out of the linguistic community, number 6 here, is that the idea here from Chomsky and some others is that there's a separate language module in the brain. And this language module has all thoughts and processes and the computations that tells the auditory circuit or the vocal circuit what to do. And it's under its command and how to process the speech and how to produce the speech. And so I've been throughout the literature, as well as did a lot of our own work in our own lab. And I argue that the evidence supports a combination of 3 hypotheses, 1, 3, and 4 here for brain differences in the size, as well as the number of packing neuron density. It's higher in the vocal learning birds. There is some differences in the presence or absence of circuits and their enhanced connectivity to the brainstem neurons that control the vocal oral facial musculature, but no evidence really of the descendant larynx. Actually, you can find the descendant larynx in some cat species, some lions, for example. We find these direct connections between vocal and motor areas in mice, and I don't really find any good evidence for a language module. So now I'm going to go through some of the evidence for this difference and explain how it came about evolutionarily. So what we found a long time ago when I was a postdoc is that when songbirds and then other species we've also seen produce their learned songs, we found that that singing behavior is associated with the rapid increase of gene expression in brain of activity-dependent genes that are responding to the action potentials in these neurons seen in white hairs, a messenger RNA for the CFOS or EGR1 gene. And it's in the very brain regions like this HVC here and area X here that are responsible for controlling the production and the learning of those vocalizations. And you don't see those activation here in silent animals who are just moving around. And here's a canary song. And what we found is that when these canaries, for every song bout that they sing, like one of those you heard, the more they sing in a half-hour period, the more rapid increase of these immediate early genes, as we call them, we see in these vocal learning nuclei, such that for this area exon nucleus, the slope of this axis here is 1:1, meaning a one-fold increase of messenger RNA product for every song bout the bird sings. And this occurs only when they sing. It doesn't require vision. It doesn't require they hear themselves sing. It doesn't require they see another bird sing. It doesn't require somatosensory feedback. It just requires that they produce the motor act of singing. So we call this motor-driven, singing-driven gene expression. And we use this as a behavioral molecular mapping tool, like an MRI kind of tool, functional MRI tool, to map communication circuits in various different species of birds. And what we found is that all species of birds that we looked at, we found these brain areas highlighted in blue that lit up when they're hearing species-specific sounds. And we now know this is their auditory pathway. And it was found in the vocal learning birds like songbirds, hummingbirds, and parrots, and the non-learning species, indicating that the vocal learning birds inherited this auditory pathway from a common ancestor of all birds, including chickens and pigeons, those without vocal learning abilities. So this was common, and it explains why auditory learning is more ubiquitous. But only in the vocal learners did we find these forebrain circuits in red and yellow here that are present in the telencephalon, or the forebrain, that are responsible for their vocal learning behavior. The yellow ones produce learned vocalizations, and the red one or respond to action imitation. They also connected with the auditory circuits. And these brain circuit has taken over a innate brainstem circuit here in gray that's found in all species producing innate vocalizations like the chicken crowing. And so what happened is, either looking at this tree here, is that either there have been 3 independent gains in these 3 red dots here of vocal learning, and each time it happened, there were exactly 7 brain structures in the similar neural network that evolved for control of this behavior. Or maybe they're so remarkably similar that we began to ask, maybe there's a common origin, and then there are 4 independent losses here. Or maybe everybody has it to various degrees, and we just haven't been able to find it here in some of these other species. Or maybe the tree is wrong, and there is a common origin and no losses, but the phylogeneticist just got it wrong. And me being a neurobiologist, I trusted the phylogeneticist to get the tree right. But I discovered when you talk to the phylogeneticist that the tree may not be right. And that then resulted in myself and others to actually sequence the whole genomes of all these species and others. And for the first time, not just look at individual genes or DNA hybridization, but look at the entire genome, and not just the gene tree, but the chromosome trees of all these species. And we got a new phylogeny here of birds back in 2014, including of vocal learners. But we rearranged the positions and the relationships of these species in the tree here, shown by these blue lines here. What about the vocal learners? Well, it turns out, actually, the vocal learners are separate. This new tree pulled hummingbirds much further away from songbirds and parrots in the family tree, indicating it's three gains, or if you want to argue a common ancestor, it has to be back here at the time of the dinosaur extinction, and then 10 or more losses amongst the water birds, mouse birds, woodpeckers, and so on. So we really think it's independent origin of these circuits. What about humans? Everybody thinks it's independent in us. We don't think we have a common ancestor with parrots, and chimpanzees lost the trait. So what about us? If these birds can evolve something similar over the last 65 million years, well, what about us? Well, we start to ask that question. And the first lesson you learn here is you take a songbird brain, a zebrafinch here, and you match it to human brain. You can fit 3,000 of these zebrafinch brains into a human brain. Brain size doesn't matter for this ability. The other is cortical folding. To increase cortical surface area doesn't matter either. What I argue is that's the presence or absence of a type of neural network that controls communication or vocal communication behavior. To get at that comparison and ask, what are the similarities? We had to reevaluate in a dogmatic view a brain organization that's been around with us for over 100 years, which argued, Lewis and Edinger back in the late 1800s, early 1900s, took Darwin's view of evolution, which was new, relatively new at the time, Aristotle's view of scala natura, thinking of evolution in a linear fashion, and thinking that evolution had a purpose, and even a religious purpose, to generate human beings. And they even threw in a little bit of racial biases here to generate European men, where they argued that the base of the human brain here, what we're calling the basal ganglia, an archistriatum structure, was first found in fish, and then passed it on to amphibians. And amphibians invented this paleostriatum structure in this basal ganglia, who then passed that on to reptiles, and reptiles invented this new structure that they called the neostriatum, neo meaning new. And then they passed that on to birds, and birds developed this light purple structure that they called the hyperstriatum. And all of these striatal areas were argued to be the basal ganglia involved in primitive innate behaviors, and that birds and lots of other vertebrates, non-mammal vertebrates, only had hippocampal cortex and olfactory cortex involved in simple cortical functions. And that this six-layered structure here in the mammal brain, they called the neocortex, being new, they argued, only in mammals, largest in European men, next largest in Asians, smaller in Africans, and smaller so in monkeys. And that was the argument at the time. And we knew this just could not be true. And when I became a new assistant professor at Duke University, I helped organize a consortium of scientists that reevaluated all the evidence, did some of our own experiments to argue that this view is totally false, and that the real view here, or what we call the Martin view, is that the avian brain has true basal ganglia structures here in purple and blue that are homologous to what you see in humans, called the globus pallidus and the striatum, and that we have this cortical territory in green in all vertebrate species, including birds. But in birds and some other vertebrates, it's nuclear in its organization, whereas in mammals, it's layered. But some of the connectivity is similar between a nuclear organization and a layered organization doing something similar. And all this is important. It's because in these cortical regions that I showed you in green in the past slide is where you find a lot of these blue, yellow, and red labeled regions, and only area X in the basal ganglia. And when we then pulled all the literature together and compared humans and non-human primates and other species, I come up with the following hypothesis, that I argue that we humans have inherited a Wernicke's area from our common ancestor with all vertebrates, and that this Wernicke's area is specialized for species-specific processing of sounds, in our case for speech. And this is why, and you can do other species as well, this is why Koko can understand how about coughing, or a dog can understand sit, siente se, roll over, and so forth, because I argue they have a Wernicke's area that can process human speech sounds and complex auditory symbols. But only the vocal learning mammals, in this case humans, I argue, have evolved the forebrain circuit in yellow and red here that has similarities to the circuits we see in the vocal learning birds. a pathway for producing the learned speech like I'm doing now, and a pathway in red that includes Broca's area involved in speech acquisition for a child learning how to produce speech. And the auditory pathway connects with this pathway in the vocal learning species we see here is one special connection. Another special connection is that this vocal learning circuit makes a direct control over the motor neurons that control the voice, the larynx muscles, and such direct projections to the motor neurons from the cortex for vocal behavior you don't find in chickens or non-human primates. By the way, when we published this, this figure here, or figures like it, was taken by some religious groups and put into religious textbooks to argue, including in Scientology, that this indicates there has to be an intelligent design, because how could you get something like this occurring multiple times in evolution with such similarities, not only amongst these birds, but even with humans? I didn't have an alternative answer to that until we came across this accidental discovery. We were trying to help a colleague discover magnetic sensing areas of the bird brain. We also discovered movement areas of the bird brain. And what we found is that when we got these birds to do controlled, learned sequential movements, like hopping in a rotating wheel in this case, the brain areas that lit up with these genes surrounded the vocal learning circuits in all vocal learning birds, songbirds, parrots, and hummingbirds, shown for songbirds in this case. And when we looked at the connectivity of these brain regions inside the song learning circuits versus the outside regions, the motor regions outside of them had similar circuits as the vocal learning circuit itself, a similar connectivity with some differences, like the direct projection I told you about earlier for the vocal pathway, not found in the motor learning pathway, and some other ones shown here with the red arrow. And this work done by a former graduate student, Giza Fienders in the lab. And guess what? Around that same time, some colleagues of mine, Steve Brown in this case, was studying the human brain and looking for the brain areas that are activated when dancers are doing choreographed, novel, learned sequential movements here in a PET imaging experiment. And shown in these red dots, lit up areas here with these blue arrows, the areas of highest activation in the human brain when we're doing learned sequential movements like dancing are directly adjacent to the vocal learning areas. And by the way, it was soon after that discovered that only vocal learning species can learn how to dance, that is humans and parrots and songbirds. And so there's some relationship there with sequential learned movement and vocal learning behavior. So this then led to what I call the motor theory of vocal learning origin. I argue that all species of vertebrates have this innate brainstem circuit in the midbrain and hindbrain controlling the production of innate sounds. All of them have a forebrain motor learning circuit that includes a part in the white arrows here for learning how to do movements, and this black arrows here for learning how to produce the learned movements. And that I argue that this motor learning circuit in the vertebrate brain, during embryonic development, hitting multiple times to control different muscle groups. And that in humans and parrots and other vocal learning birds, I argue that there's one more duplication, a whole brain pathway duplication, like a gene duplication of this motor learning circuit, and that it then took over control of the innate brainstem circuit and put those two together. You get a novel vocal learning circuit that evolves from a deep homology with the surrounding motor learning pathway that controls other things besides vocalizations. So if this were true, then we would expect the genes that function inside these vocal learning circuits would be similar to the surrounding areas, except for those that control some differences in the connectivity, or maybe differences in plasticity of how these circuits function. And so that brings me to the third part of my talk here about the genes. And so we started to investigate what's going on at the genetic level. We said, if the behavior, if the circuitry, and I didn't get into the physiology, but some of the physiology is similar between these birds and humans, if all of that is similar, then maybe the underlying genetics also evolved in a convergent way. And so to test whether that's true or not, what we did is we dissected out the vocal learning brain regions using a laser microscope dissection on tissue sections of these brains and the surrounding areas, and profiled them to what used to be used a lot called oligonucleotide microarrays, where there are thousands of genes that you can test for their expression on a glass slide. The Allen Institute for Brain Science was doing something similar to 3,700 human samples that they got from 6 postmortem brains, people who donated their organs to science, several hundred from 6 monkey brains, including motor areas where we would expect to find vocal learning circuits, and profile them to microarrays. And students and postdocs in my lab, like Andres Fenning, Irina Hara, and Osceola Whitney, who did a lot of the wet lab work and also the computational analysis, analyzed this data. They all have their own labs now. And more recent work from Gregory Jedman in my lab, who's using a new technology called RNA-seq to measure the expression of all genes that are expressed in these cells. And what we found is that when we look at the gene regulation in these brain areas between songbirds and humans using new computational approaches, looking at 7,000 or more genes, we find that genes that are specialized in their regulation in the song nucleus of the HVSC of songbirds match genes that are specialized in the human laryngomotor cortex more than anywhere else in the human brain. In the cortex, the basal ganglia, the cerebellum, the closest and the most strongest match is laryngomotor cortex, and particularly layer 2 and 3 neurons, cell types of that cortical region. This RA song nucleus that makes that connection out of the brain matches another neuron type in the laryngomotor cortex, whereas this part of the anterior striatum of the human brain and the songbird brain match each other, and was predictive, where subsequent colleagues of mine have gone to, and then looked at human brains, us, that is people, in scanners, and asked what is the brain area of the striatum that shows the highest activation when we're learning how to do novel speech sounds, and it's this region where we find specialized gene regulation, up or down regulation of the gene product compared to the surrounding circuits. And we did not find such specialized changes in these genes in the brains of vocal non-learning birds like quails or pigeons, or non-human primates like macaques or marmosets. And then we start to look at the individual genes, and we can, each row here is an individual gene, and we're looking at one of those brain areas, the RA song nucleus of birds and laryngomotor cortex of humans, and blue is downregulated and red is upregulated. And what we can see for the songbirds, the parrots, and the hummingbirds, these 3 species here, we see that the pattern of gene regulation for these 55 genes is very similar to what we see in the human speech motor cortex here. And that pattern we don't find in the doves or the quails or in other non-speech areas of the human brain, and not shown here for non-human primates. And what's striking is a lot of these genes are turned down or turned off. They're blue here. And a lot of those that are turned down are genes like what we call SLIT1 or GAP43. These are genes involved in forming neural connections during development and maintaining them as adults. So we think those could be responsible for making a difference in the speech circuits. And some of them, like SLIT1, are also, when mutated in humans, causes speech deficits as well as speech disorders in autism. Other genes are upregulated, like parvalbumin here, and these genes are involved in neuroprotection. They protect neurons from getting toxicity from too much calcium use or glutamate use. And I think this evolved because vocal learners tend to be the most vocal proliferous animals out there, like us humans. We vocalize a lot more than any other primate. Songbirds sing a lot more than any other birds out there who aren't vocal learners. And so I think once we evolve the circuits, we use it a lot, and we can use it so much that we can drive toxicity. So we need these molecules to get rid of that. We validated some of these by a method we call in situ hybridization, like I showed you before, where we can actually measure the expression of the genes inside the brain. And we can prove that they are downregulated, like shown here in the 3 vocal learning bird groups. We don't find that in the non-learners. And in humans, the white areas here, we can find it downregulated. There are actually 2 circuits we find in humans. And guess what? 2 in parrots as well, compared to the songbirds and hummingbirds. And we think this gives humans and parrots more advanced abilities to produce imitated sounds than other species. These areas where you find the specialized regulation of genes actually correspond to the areas of the brain that a colleague of mine, Eddie Chang at UCSF, looking at human patients when they're removing tumors in epileptic regions to avoid speech regions. They go in there with the electrodes and they poke around in the brain and ask the patients to speak under surgery. And they find these 2 hotspots in red here of highest activation in the human brain when speaking. Those 2 hotspots correspond exactly to where we see the specialized regulation of genes here. And so now what we're going to do is investigate the function of these genes. And to do so, we need to study them in cultured cells and so forth. And here's one of those genes, SLIT1 I said before. But what we discovered is that the genome assemblies that the genome biologist did to put these genes together had lots of gaps in them. And in blue here shows low quality sequence, such that we had missing parts of genes or even entire genes that were missing. They were incorrectly assembled together. The puzzles of the DNA were put together incorrectly, and which caused meaningful biological regions like repetitive regions of the genome to be misassembled. This was causing students in my lab to take many months to correct and clone these genes. Imagine when you have 55 of them per brain region, and you're unknowingly working with artifactual gene structure and sequence, unknowingly making false conclusions about the biology. And this really troubled me for practical and moral reasons. And because of that, and because I started to get heavily involved in genomics and became chair of what we now call the Vertebrate Genomes Project. And we set our goal to have an ambitious mission with the goal of to generate at least one high quality, error-free, near gapless, chromosomal level, and phased haplotypes, maternal and paternal chromosomes, and annotate all the genes in those assemblies from all extent 70,000 vertebrate species, and utilize those genomes to address fundamental questions in biology, disease, as well as conservation, given the current mass extinction we're undergoing now. And we found a formula that is generating real high quality genomic data. It's using what we call long read sequencing, molecules that are 10,000 base pairs or bigger, and linked read to bring those long reads together. And three different approaches here we call link reads, optical maps, and then 3D chromosome interaction information, using these three technologies to take those long reads and string them together into entire chromosomes, fill in any missing gaps we can find with some of these reads, correct some of the base call errors, and then apply this technique with different size insert molecules going from left to right here in various species, including some vocal learning bats, some of my favorite vocal learning birds like the zebrafinch here, and then ask the questions about the genes that we want to study. And long and behold, SLIT1, shown in green here, is now in one long sequence with 0 gaps here. And we found in some of the gaps in the old assembly, we're getting the wrong amino acid predicted sequence here because the gaps are causing problems. And so now we have the right protein coding sequence, and we also have the right regulatory regions. And to make a long story short, recent work in my lab, this is unpublished stuff now that we're working on, with these new genome assemblies, we're discovering that the vocal learning species in some regulatory regions of the genes that are specialized inside the circuits have mutations in them, shown here in blue. And some of the mutations are the same, and some of them are different. And they have mutations in the regulatory region upstream of this NeuroD6 transcription factor. And this mutation converts a supposed non-functional sequence to a sequence that allows a transcription factor to bind, in this case, the androgen receptor. That receptor binding is suppressing the expression of this transcription factor. And when you suppress the expression of that transcription factor, then it can no longer upregulate the gene SLIT1 in the vocal learning species. But we do want the gene SLIT1 to be upregulated in other brain circuits. And so what we're finding is that in the non-vocal circuits of the vocal learning species, we're finding that those regions with these novel mutations have histone coated around them so that these repressors can't bind to them and not prevent the downregulation of this gene in the non-vocal circuits. And so that's how we think a mechanism is going on to cause the genes to be specialized. Why specialize the gene in the first place? So some of these genes, I said, are involved in neuroconnectivity, and the specialized connection you see in the human and other vocal learners is from this part of the brain here, this motor cortex region involved in speech, making a direct and indirect projections to the neurons here in yellow, nucleus ambiguous, that controls the larynx, and you don't see that in a monkey, all right? So why have the downregulation of the gene here? Well, we think that these neurons up here, shown now in green, they make high levels of the SLIT1 gene in the non-human primates. And we think that high level of SLIT1, we know when SLIT binds to its receptor, which is in these motor neurons that control the vocal organs, when it binds, it says it don't connect to me, go away from me. It's a repelling function. It says, please don't touch me, basically. Whereas in the vocal learning species, the slit is turned down. And we think then that allows the axons from these vocalizations, these speech motor neurons, to now form a direct connection, even when the receptor is there, to not be repelled and have the cortex have direct control of the muscles that then control speech. And so if this were true, it would mean that we could take a songbird or a vocal learning species and upregulate SLIT1 and prevent the direct connection from forming and see if we can block vocal learning. Or maybe we can downregulate it in a species that can't imitate, make an indirect projection direct, take over an existing motor learning circuit, and get this chicken to actually imitate something. And we would love to do it in a chicken. We would love to do it in a non-human primate, but that's hard. The genetic tools aren't there so much for those species, but they are there for the mouse. And so we began to study mice and ask the question, what is the mice circuit like? And all of us scientists were assuming that mice were not vocal learners. We're basically vocal non-learners. And so now I'm going to tell you about ending off with hypothesis testing in mice, leading to a new idea of how vocal learning circuits evolve. Because it was discovered around that time that, actually even before we began some of this, is that mice have these ultrasonic vocalizations, particularly the males, that sound something like this. I'll play that again. All right. And they use these vocalizations to chase after females. And the more complex the syllables, the more the females like them. And so this to us sounded like songbird songs. And this prompted us to think, are these mice vocal learners and we just missed it all these years? And even different brothers from the same parents, mouse 1, 2, and 3 here, will sing different compositions of 3 different syllable types here, indicating there is something that is can be different from one animal to the next. And so there were multiple laboratories that got together, and some found that there is a small level of vocal plasticity. Others argued nothing at all, but none of these studies argued that mice are vocal learners, including work from students and postdocs in my own lab here. And so it's just an interesting variety of sounds that are innate, basically. But there are social context differences. There are meaningful differences, because if you place a female in a Y-arm maze and ask her which songs does she like from these ultrasonic songs of these mice, she will spend more time next to what we call the sexy songs, the more varied types of sounds and the more simple ones than that they sing. What about the forebrain? If we get these mice to vocalize like we did for the birds, we find that they actually have forebrain regions that light up like we see in birds and humans. That was a surprise. And when we place tracers in the muscles and ask what brain regions they go back to, the tracers go back to the same area that we see light up with vocalizing in these mice, and particularly these layer 5 neurons here. And they're distinct neurons. And when we place tracers in those layer 5 neurons and ask where they connect to, they connect directly, shown by these black lines here, to the motor neurons here in red on this side, or brown here, that control the vocal behavior muscles. And so, but a difference here with mice compared to humans and songbirds is that this is a very sparse connection. It's like 1 to 3 axons per motor neuron. And to compare here we have a songbird, here we have human, many axons coming down from the forebrain, thousands, hundreds and to thousands, connecting to the motor neurons here for vocalizations in songbirds and humans, post-mortem brains here in humans, where you can label degenerating axons from a patient who had stroke in the brain, in the motor cortex, where again, shown here for the mouse, only a little bit. So what about the genes themselves, the downregulated ones, for example? We don't find them downregulated here in the mouse brain. And so this led us to what we call the continuum hypothesis, where we argue that it's not a black or white world, but it's a step function here, where you got species like lizards who don't vocalize at all right? So there's no vocal learning to happen at all, to ones like chicken and mice, who we might say are limited with limited vocal plasticity, can do some small modifications, to moderate vocal learners, to complex ones like the bats and the songbirds, who can imitate lots of different sounds to the high vocal learners like the parrots and humans who are the more advanced vocal learners. And that as you go up this stepwise continuum, you find fewer and fewer species with those abilities until you get to humans. And what this means is that to manipulate these circuits with these genes, it may be not a problem of trying to induce or get a whole brain pathway to duplicate, but to modify an existing sparse connection to become strong and maybe get these mice to imitate. And that's what we're trying to do going forward in the future. And we have some preliminary data shown here where we were able to create transgenic mice with the SLIT1 gene downregulated like it is in the human brain, but in this case, throughout the motor cortex, the whole cortex. And these mice are alive, they're well, these mice spontaneously sing these more complex syllables more often compared to their wild type or heterozygous controls. So these syllable types would grow with. This was recent work from a postdoc in my lab. And we're, if you invite me from another year from now, I can tell you whether these mice imitate or not. I don't think so. We probably have to change more than one gene, but we're trying to get there. What about other genes in this pathway? It turns out one of those genes that affects SLIT1 is FOXP2. And some of you may have heard about FOXP2. It's a gene when mutated in humans causes a very well-known and prevalent speech deficit. And here is a child with a FOXP2 mutation of this transcription factor.
May I ask
your name?
She's trying to say Laura.
Where do you live, Laura?
She's trying to say Sheffield.
And how old are you?
4.
Your name?
4. So Laura is a 4-year-old child who at that time lived in Sheffield in the UK, has a heterozygous mutation of this transcription factor, and is pretty good with auditory learning and auditory comprehension. You can understand she understands pretty well the person speaking to her, but she has difficulty producing learned speech. The motor output is where the problem exists. She can ride a bike, she can walk just fine, she can have other kinds of movement learning, but speech is the most affected motor learning in this case. And her siblings who don't have this mutation when they were 4 years old were doing just fine like a regular normal school child. And so we actually took Laura's mutation and put it into a mouse genome and found an impact. What happened is that wild type mice, like her siblings, when you take these males and put females in front of them, they switch to these more complex syllable types that the females like. However, the mice with the FOXP2 mutation did not switch to these more complex syllable types. With the underlying deficit here, we think they have difficulty sequencing complex syllable types together, like sequences of phonemes to make words. And what about the neurons in the brain that synapse down to the motor neurons for the vocal behavior? They are there in the mice with the FOXP2 mutation. However, unlike the wild type mice where you have these neurons sort of concentrated here, shown by the black bars, in the primary motor cortex, in the FOXP2 mutant mice, they're more spread out. And so we think FOXP2 is also necessary to co-localize these motor neurons in a localized area of the brain to synapse down onto the motor neurons. And so what we think is going on here is that these mice have a limited ability and this FOXP2 mutation was used by non-vocal learning species like primates or the non-vocal learning birds for vocal behavior even before speech evolved. But when speech evolved, it became used in a more complex fashion. So to summarize here, what I'm arguing is that we can learn a lot from studying species more distantly related to us, even for traits like speech. And that we've learned so much about the songbird vocal pathway here, shown on your right, down to the individual cell types and neurons and specific connections to specific dendrites and axons, and all the neurons involved in the brains, in the forebrain, and the brainstem circuit here, and the muscles, and how they control sounds. And with this comparisons at the molecular and other levels, we can now say what cell types in the human brain, in the songbird brain, we can predict what the speech circuits would work like. And so now the songbird model, even though 300 million years removed from a common ancestor with us, is being predictive of how these circuits evolve because their convergence into a similar direction, not everything is the same, but a lot is the same. And so I hope now, and I can see that the human neurobiology world is taking notice of these findings and learning from them to understand human speech function and disorders. And what about outside of humans? Well, if humans and the vocal learning birds evolve similar brain regions with similar connections to the peripheral vocal organs for speech and song, who's to say another 500,000 or several million years from now, some other mammals or even some reptiles or amphibians, heck, a fish, would evolve these similar types of brain regions out of their motor learning pathways by a brain pathway duplication, enhance them with these genes, and then get vocal learning. And so by making all these comparisons, I think one day we really will understand what makes the difference between us and our close relatives who can't imitate these sounds. And if we want to do, not only repair our own brain circuits after stroke and so forth for speech, but also try to enhance such circuits in other species that either have them at a rudimentary degree or even advanced degrees. And so I will summarize here by saying the high take-home level message here is that complex behaviors can evolve multiple times from deeply homologous but diverse brain circuits, cells, and genes. A trait like spoken language, as complex as you may think it can, as it is, actually can be understood from studies in distantly related animals. We still need to study humans to do that. And convergent evolution of mechanisms for a trait is associated with similarities and disorders of that trait, like autism or FOXP2 different mutations. And so I thank the people along the way who's led some of these individual projects. This is my team back when we first started. We're now actually even bigger than this at Rockefeller University. Collaborators I had from around the world for these various projects, students who have been in my lab before that now have their own labs or working in other capacities, as well as funding from the federal government, from NIH, from NSF, from my Howard Hughes position, the Packard Foundation, the Allen Institute for Brain Science, as well as funding from the universities themselves. We depend upon university support like the Rockefeller University and Duke University. So I will go ahead and stop there and say thank you for your attention and take questions.
Awesome. Thank you so much, Dr. Jarvis. That was definitely fantastic.
You're welcome.
We're having plenty of conversations and questions happening in the chat. So I think we're ready for our question and answer section.
Okay.
Thanks,
okay.
Dr. Jarvis. Jen, do we see any folks who are raising their hands that we want to start with? Should we go to Brittany?
Just want to say hi, everyone. It's nice to be in a room with you. And thank you to MFA and thank you to Dr. Jarvis. So I have a bit of a pop culture question. I and my AP biology students are obsessed with Bunny the dog. And if you are not familiar with Bunny the dog, and I can tell the people in the frame who are because they're reacting very positively, Bunny the dog is a dog that, according to TikTok and social media, has about 70 to 80 buttons and can push and communicate, seemingly communicate with their owner, even on abstract concepts like whys and things out of the room, and even recently in a video, a sense of self. And so I was just curious if you think that's a scam or not, if you would consider that communication, if that aligns with your research or not, and just any opinions that you have about that form of seeming communication.
Yeah, with the ability of the, let's say the video world and picture world to make things so realistic, seem so realistic and being fake, it's a good valid question to ask. You know, is this fake or not? I actually haven't heard Bunny the dog, so this is the first time I'm hearing it. I have to go look into this myself to be able to give you a well-informed answer. And if this is happening, I want to go see this dog myself. But with that said, just based upon our work in general, I can make some predictions. We often underestimate what some other animals can do, and then sometimes we might overestimate. And we underestimate if we don't really understand them. And so with studies with Koko, for example, I said several thousand. Actually, Koko could understand 7,000 words before she had died. That's a lot of words to understand, whereas by an adult human is at 20,000 or more, right? And so so I think if we can train some animals to respond and understand our speech and get them to do non-vocal motor output with gesturing with the hands like Koko does, or with the dog using the paws, I do believe it is possible to train some dogs to actually respond to have a conversation, a mini conversation beyond simple yes or no answers. But can they have a complex conversation with a lot of grammar and syntax? I don't know the answer to that. I think once you evolve the speech circuit, having the speech circuit helps the comprehension work. If you don't have that speech circuit in a dog or Koko, I think there is some limitation there. So I don't think they're going to have a conversation like you see in some cartoons.
Great. Thank you so much, Brittany and Dr. Jarvis. Pete is raising his hand. Pete?
Thanks, Isma. And thank you, Dr. Jarvis. I do have a question. It's of a personal nature. I have a stutter. I grew up with a stutter. And it's kind of gone away. It's still there, but when you go to speech therapy as a kid in the 90s, you learn how to avoid the stutter.
Mm-hmm.
As you move on in your life, I had therapies that dealt with the anxiety of the speech itself. We never ever got into the genetics or the possible genetics of this. And when you briefly said at the end there, other speech abnormalities, other scientists studying that, I wonder, we train ourselves very hard to change our speech pattern because of the stutter. So, is there any research into stuttering being part of this genetic way of different parts of the brain and how they communicate?
Mm-hmm. Yes, good question. And actually, we've published several papers ourselves where we actually discovered stuttering can occur in vocal learning birds, and some of the reasons why. One happens what they call neurogenic stuttering, which is stuttering that occurs as a result of brain injury. And we found that what happens, unlike humans, bird brains can regenerate neurons a lot easier than mammal brains. And we found that when these new neurons are regenerating in the basal ganglia structure, as I mentioned before, the birds start to stutter. And then they can overcome the stuttering after the neurons are finished making new connections. And so what we think is going on there is that the stuttering is occurring because there's a timing delay that needs to be overcome in connections in certain parts of the brain. And a lot of therapies focused around stuttering is actually focused on timing of speech and coordinating that timing of what you produce and what you hear, getting those two things lined up. And so we think we can learn from these birds on how to not only induce, but also how to repair stuttering for this region. And so I think to broaden out further is that with the ability of spoken language comes certain things that can also be a problem with spoken language that comes about. And we can learn about how to overcome those when even studying in these other species.
Thanks for that. Great.
So good. Yes, thank you.
That was great. We have a question in the chat from Steve, and Steve would like to know, what is the distinction between a non-vocal learner and a non-learning vocal ability? Does it involve the potential to learn or the genetic physical structure?
Yeah, so there isn't a different. I just interchanged the words vocal learner and a non-vocal learner. So the same thing. And what makes the difference between them and the vocal learners is a combination of the genetic and the actual structure in the brain. So it's the genetic differences that are driving the structural differences in the brain.
Great, thank you, Steve. We have another question in the chat. This one is from Kit. Kit would like to know, how these areas of the brain differ in people who are deaf and use sign language to communicate versus those that show speech and song and verbal. Do the areas of the brain that show speech and song and verbal languages get rewired to match when they're using their hands to communicate versus when they are doing other actions with their hands?
Yeah, this is an area of debate, and I stand on one side of that debate that I put into my article. One view is that that the areas coming from more this language module belief is that the areas that control speech and the areas that control sign communication are the same brain areas used in different ways. And another view that I have is they're not the same brain area. Like I said, for the dancers, their brain areas that are next to each other. You have parallel circuits, one next to another, that control different body parts, basically, including communicating with the voice or communicating with the hands. And the reason why I think some have argued that they're the same brain areas, when you're using functional MRI, it's hard to get good resolution of adjacent brain regions next to each other. But when the patient is undergoing surgery and you put electrodes in those brain areas, you'll find that these neurons are actually near each other, but they're still more distinct than similar in terms of sign gesturing and speech. Nevertheless, I think there is a relationship. And you'll see as I'm gesturing, sometimes as you're asking questions, I mean, as I'm speaking, I'm gesturing with my hands here. Because why? I think the brain areas that gave rise to speech evolved by a brain pathway duplications of the brain areas that were controlling gesturing, like we see in Koko, who has that ability, but does not have the speech ability.
Thank you. We have a question from Susan in the chat, and she wants to build on Pete's question and ask if there are other studies that are being done with persons with verbal disabilities and autism spectrum disorders.
Yes, so we and others as well, but we're gearing up for a project, applying for some grant funding to look at autism, because we found some of those genes that are specialized in speech circuit, like I said, also when mutated or has certain substitutions in them, can cause certain autistic deficits. The issue here is that with autism, there's so many different sub-traits associated with autism. Like I said in my beginning slide, like language itself, it consists of sub-traits. And so what we think scientists need to do is break them down into different subcategories if you're going to study them properly. And we are going to we're going to sequence the genomes of children and their parents, because we're looking to see if these are de novo genetic changes in the child, or it's a combination of genetic changes they inherited from the parents. So you can sequence the genomes of parents and their children, and the affected siblings and the non-affected siblings, and ask, do we see any genetic changes in some of these genes that are specialized in these speech circuits for children who have speech deficits associated with their autism? And we think the answer is going to be yes. And with that, then, if we can find those specific genetic differences, then we can recapitulate them in the mice, in the songbirds, and figure out how to actually repair them.
Great, thank you. I'm going to go to a question from Emily, but also connects back to a number of questions that we saw earlier in the chat regarding cetaceans and the research around, if there's any research around their vocalizations. So Emily is asking that she's heard of studies showing that some parrots, bats, and cetaceans have vocal names that they use to refer to other individuals in their social groups. Do you think there are specific circuits would require this ability, or possible links between vocal learning and higher order processing? And then just generally, a lot of folks were asking about research that was done, if you can share research that was done on whales and dolphins around vocalization, which is a very broad question.
Yes, actually it's hard to do research on whales and dolphins because they're so big and they're in the ocean, right? And you do have some in SeaWorld like habits, but still, even it's hard to study such big animals. Nevertheless, they've been studied, and they have some of these males. They have some of these long songs that last 20 minutes, And they have big brains. Their brains are bigger than ours, so they can store more information. And they'll have these songs that go from one generation to another and have dialects in different languages and different oceans and so forth. So to the question about the brain circuits and naming, no one's actually discovered what the vocal learning circuits in dolphins are like, because no one's looked, and it's hard to know. But with these specialized genes we found in humans and songbirds, we're now getting post-mortem dolphin brains from strandings, dolphins who naturally die. And we're looking at their motor cortex region with these genetic markers to see if we can find their vocal learning brain regions. And we don't have the answer yet, but I have a graduate student in the lab doing that with dolphin brains. With that said, those who have these signature whistles, they call, where they name each other according to different whistle sounds, like we name each other, actually dolphins do this. And my guess, I doubt that's going to be a separate brain circuit one day when we do MRI on them. I think we'll find out that it's the same brain circuit like we have for speech, just being used in different ways that you don't see in other vocal learning species.
So fascinating. I think we all have a million more questions about the vocalizations of
yeah, I see the chat is lighting up.
Yeah,
right. I'll try to answer quicker now so I can get to all your questions. All right.
I wanted to go to some questions around the FOXP2 gene.
Mm-hmm.
So Jerry is asking, does the expression of the FOXP2 gene differ between females and male animals?
Good question. Not in humans, although in humans, boys have twice as high probability of getting a speech autistic deficit disorder as women do. So we think maybe not FOXP2, but something on the X and Y chromosomes is impacting our vocal communication circuits. And because boys have only one copy of the X, if you get some type of gene that has some type of change on the X chromosome and you don't have the other half, the other X to compensate it, then you're going to have a deficit. But FOXP2 is not one of those because it's not on the sex chromosomes. We think that though, what's interesting in mice is that these produce these but these innate long sequences of vocalizations, the females don't. But if you get damage to the olfactory bulb in female, they'll go ahead and produce it as well. In a parallel situation, in songbirds, lots of songbirds away from the equator, not those near the equator, but the further you get away from the equator, the more likely you're going to find songbirds where only the males sing and the females don't, all right? But what happened there is that there's some evolutionary mechanism that evolved the loss of vocal learning abilities in females rather than the other way around. So both sexes evolved at first and females lost it. And you can prevent that loss from happening if you give those female songbirds an extra dose of estrogen soon after they're born, after they're hatched. You give them an extra dose of estrogen for 1 week, they grow up like the males and sing a learned song. So there's some remarkable sex-specific things going on here. Now, when it comes to humans, it turns out not only for autistic deficits, but it is true, and I'm writing a review on this now, that actually women do have more advanced speech and language skills compared to men on average. There's a lot of overlap, of course, but you know, the mean is skewing towards women. And this is not only thinking because of the X chromosome, because something about in women, there's more corpus callosum connections between the two hemispheres in women compared to men. Why that's the case, we have no idea. But what happens is because when brain damage occurs in one hemisphere for speech, in women, the other hemisphere can take over easier than it does in men. And why that's the case, we have no idea, but it's still a sex difference. So I took the liberty to answer a lot of things about sex differences with that one question. Yeah.
That was great. Thank you so much. Era has a great question. I want to come back to it. Era, I just want you to know that I see it. There were a flurry of questions earlier that I want to come back to first around dance and language. And I had the opportunity to hear you speak before, and I know that you had an interesting journey of deciding between dance and neuroscience. But folks want to know, does the FOXP2 gene make birds good dancers? What is the biological definition of dance? And are dancers more adept at language? So sort of three questions around birds and dance, what is dance, and the connection between dance and vocalization.
All right, let's see if I can answer all those questions quickly. And with this group appreciating, I grew up in New York City in a dancing neighborhood in Corona, Queens, and went to elementary school out there. And then lived in the Bronx, went to school there as well in Burnside Avenue area, went to high school of performing arts and became a dancer. I majored in dance, on 46th Street. And I was then trying to decide a career in dance with Alvin Ailey because they were asking me to audition for their dance company or go into science. I chose science because I felt I could have a better impact on the world that way. But now there being a relationship between dance and speech, I'm back where I started. And I haven't stopped dancing either. Instead of doing ballet and jazz and so forth, I'm now doing a lot of salsa dancing and still performing that. And so, so how to bring all that together? What I think happened is that when the vocal learning circuit evolved outside of the motor areas that control other body parts, that in order to involve of speech, we need to have a rapid integration to the sounds we hear to the motor circuits that control the sound production. And so with this auditory motor integration being very tight and specialized in the vocal learning species, I think it contaminated the surrounding brain circuits that are now that contamination allows us to learn how to dance, to move our body muscles to the rhythmic beat of sounds. You can't get that in a dog, you can't get that in a chicken, but you can get it in a parrot, in a human. And so what are the genetic changes behind that? We're finding that some of the genes that are specialized in the vocal learning circuit, not all of them, but some of them are specialized in their regulation in the surrounding brain regions, like SLIT1. But FOXP2 is not one of them. So I don't think FOXP2 is giving us that ability to dance, but I do think some of the other genes that are functioning in parallel with it are allowing us to have that ability. Now, when I say this, sometimes some people say, I can't dance, right? I'm no good at it, I'm arrhythmic. It is true, there's some people out there that are really, truly arrhythmic, but they're pretty rare. If you compare yourself to a dog or to a chicken, I guarantee you, if you're human and you have good speech skills, you can learn to dance better than your dog. All right, with that said, all right, does dancing have the ability, is dance skills, music skills, if you want to say as well, and speech skills all connected to each other? The answer to that is not known yet. You got to send some students our way who want to study this and say, join the Jarvis lab to study that question. But there's some anecdotal findings leading in this direction that indicates if you're a good musician, all right, you have better speech and language skills. But we don't know if you're a good dancer, do you have better speech and language skills? If all of those go hand in hand, right? One day you'll be putting music and dance into your curriculum as a more academic exercise than a simply artistic exercise.
That was great. So many people are commenting about dance and their love of dance, and that they can't dance or they can dance, and that we should conclude with a dance party. So maybe we come back to that. So I want to come back to Era's question, which is a really compelling and powerful one. She asks, why did it take us until the 1990s to challenge a racist view of the brain? Was it not a priority to challenge prior to that, or was the view just so thoroughly accepted?
Two answers to that. By the way, I see Jean said she teaches at PS 92. My cousin went there in Corona. So nice place. And one of my colleagues who works with me now in my lab also went there, who's also into dancing. So, so the question was about the racism. I think two reasons, all right? One reason is that a lot of scientists don't even realize that some of these terminology in our understanding of brain organization became contaminated with a racist view. And so they don't realize that the history exists. And the others, some do, and they're okay with it. Because look at our current political climate and culture now, that I think, you know, as Biden has said, that some, you know, some people can bring out our angels and they can bring out the worst in us. And I'm surprised, even in education or in the academic science community, whether it's implicit or unconscious views that people can have, some people believe there is a genetic difference in African Americans or Africans generally that makes them so-called less smarter. They actually really believe that. And so it's not a lot, but it's enough to keep views lasting around a long time. And talking about the 1990s, when I was a graduate student, you know, I was born 1965, and my mother tells me, you know, when, you know, the time she was walking down the street with me in her hand and people telling her to go home because Martin Luther King had just been killed, it's dangerous to be out on the streets and so forth. That's in my own lifetime. And so 30 years from the 1960s to the 1990s, I don't think the human brain changed genetically. I think the brain is the same. It takes multiple generations for the genetic changes to occur. And I think we need a lot more cultural changing and new knowledge to get rid of this racial disease in our society.
Yes, thank you so much.
Yeah,
Kendra says you're preaching to the choir. Absolutely. Such important work that you're doing on a variety of topics, and we really, really appreciate your work and efforts toward equity in science as well. We do have a few more questions. We do have a few more minutes for questions. So if anyone would like to use the hand raise feature or type a question into the chat, there may have been a few from earlier on that we missed. And if you'd like to put those in the chat now or use the raise hand feature, that would be great. So we'll give folks a second.
Yeah, I thought I saw one that I didn't answer.
Yeah, there are a couple of questions earlier.
Yeah, hear about deafening, right.
Jerry wanted to know, just a quick question, if birds have both a Broca and a Wernicke's area.
Yeah, so the answer is yes. So the Wernicke's area, I think, is evolved from a culture in everybody, and the vocal learning birds, they have a Broca's-like region, but of all the regions that are similar for speech between songbirds and humans or parrots and humans, the Broca's region is the most different. So it's the most specialized in human, and it's the most specialized in these birds. And I think that's where the real difference in complexity is happening. And I see a question here from Kit about the becoming deaf and rewiring of the circuits. I know we answered something about this, but that question is making me think something more about this, about when you become deaf and when you become blind. All right. So Helen Neville and others have shown, and we've seen this also in birds in some preliminary data, is that if you are deaf, whoops, I did something here. Let me escape that. Can you hear me still?
Yes.
Great, okay. All right, if you are deaf, if you become deaf as a young child or born deaf before puberty, then what happens is that the auditory cortex sitting at the sides here get taken over by other sensory cortices, like the somatosensory cortex and particularly the visual cortex. And so then you have a greater visual expansion of cortical usage, and you can have actually heightened visual awareness and visual processing compared to normal hearing people. So you're better at vision, basically. Vice versa if you are born blind. But after puberty, if you become deaf or blind, you don't get that cortical expansion takeover. So the brain area that was used for hearing or seeing will shrink down somewhat, not not by a lot, but it will still shrink down, but it just won't be used that much and not be in takeover. There's less plasticity. So there's something about that transition of puberty when the hormonal changes for testosterone and estrogen and other things set in, they more hardwire the brain such that even for damage to the brain, it's hard to be flexible and change things.
That's fascinating. So we have a couple more questions in the chat. Alex would like to know if there are other areas of research in other sciences, such as evolutionary psychology or anthropology, that might help to either clarify your research or inform a more thorough application of your findings.
Yes, so evolutionary psychology or anthropology, what I think needs to happen for them to clarify my research, as well as to clarify the two, in a reciprocal manner. And when I say my research, I'm thinking about neuroscience, I'm thinking about genetics. Is that they need to have a more marriage between the fields, and so whether it be physical anthropology or social anthropology and so forth, we need to get more at the biological basis of social anthropology or psychology and evolutionary psychology as well, to understand what's the interaction between cultural evolution, environmental influences on our behavior culturally that's learned, and the underlying genetics that constrain those cultural differences. And what I think we don't have enough now, whether you talk about language or you're talking about dance or you're talking about mathematical calculations, we don't have enough of an integration between those fields to be able to, I think, advance them far enough as they could be advanced.
Great, thank you. So we're probably going to wrap up with this question, and then Dr. Jarvis leading us in a dance party. Several people would like to know about the connection between birds and dinosaurs, and if you think that some dinosaurs may have had vocalization capabilities or were vocal learners, if there's any evidence to suggest that.
Yes. So it's pretty convincing now, looking at whole genomes and figuring out who's related to who, that birds are basically dinosaurs, right? They're reptiles, and they're a specialized dinosaurial type of reptile, the only ones that survived the last mass extinction. And we think the reason why they survived is that there are few species of birds that were generalists, that were able to live in water, on the coastline, in forest and desert regions. They were genetically adapted to be generalists and survive in multiple types of environments. And then they evolved and differentiated into all the other kinds of species that we have now, what we call modern birds. We call neo-aves, which make up 85% of all bird species, 95%. That includes the parrots, the songbirds, the hummingbirds, the chickens, the woodpeckers, the cuckoos, and so forth. Then the ancient birds, like the chickens, right? The chickens are actually closer to dinosaurs than the songbird is, right? The chickens and the emu and so forth, they're the more ancient-like birds, retaining some more of those ancestral traits. And so, so we've, in the beginning of my talk, I told you about the brain cell type similarities between humans and birds, or between mammals and birds. Well, it turns out we started looking at those cell types in reptile brains, and guess what? They're all there that we see in the birds. They're somewhat organized differently in different parts of crocodile and some lizards and turtle brains, but they are there. And so to me, that does mean they had to be there in a dinosaur. And we're writing a paper now, what a dinosaur brain organization looked like. All right. And it also means they had to have the capacity to evolve vocal learning circuits like the birds did. And whether they were talking dinosaurs, I don't know the answer to that. But I think we get to learn the culture and the output of these birds more deeply. We can figure out one day and predict one day whether there are some dinosaurs out there, whether it be a Tyrannosaurus Rex or some other species that had some kind of vocal learning abilities. But I wouldn't be surprised now. And just to add to that, the common ancestor with parrots goes back to falcons 50 million years ago, all right? And so it had to be about 50 million years ago that parrots evolved vocal learning. And it's been with them for a long time. Humans, it can't be no more than 500,000 to 1 million years ago.
So on that note, I think there are a lot of folks who agree that that all future sci-fi films are going to have to be reevaluated with dancing dinosaurs and feathered dinosaurs and singing dinosaurs, and that Pixar will be probably right at the cusp of changing how we view vocalization and dance in dinosaurs. So we want to thank you so much. This has been such a fabulous talk. There are a million more questions, and the conversation will certainly continue. So thank you again to Dr. Jarvis for taking the time to be here with us. Good luck in your continued work and efforts. We look forward to seeing how that all unfolds over the next year. And we want to thank everyone who's been here tonight. So thanks again, Dr. Jarvis. And
you're welcome. And I see all the thank yous coming through the chat.
Yes,
I really appreciate that.
We loved
Yes.
it. We loved it.
Yes.
It was so wonderful. Thank you so much.
Okay. Bye, everybody.